Hello, I am Amelia, and today we will be talking about mitosis, the process of asexually reproducing. I made a little joke on it, which I will be putting somewhere around here. So mitosis is when your cells, either because they're dying or you need new ones because like you cut yourself or something like that. It's not the Band-Aid that's the magical thing. It's you that's the magical thing. But Band-Aids probably do have some magic in them. Placebo. So cells, when they reproduce, have to be identical. Otherwise weird stuff happens. If they do too much, then that results in cancer, because your body's just like, everyone split up and make more. I'm tired, okay? Mitosis takes up only 10% of the cell cycle, and interphase is the other 90%. Because again, too much mitosis makes cancer, which is bad. The nucleus holds the DNA, and here are the stages of mitosis. Prophase, which is beginning, and things are starting to duplicate. In metaphase, the chromosomes are lining up in the middle of the cell, and the nucleus has decided to leave. Anaphase, chromosomes are being pulled to the opposite poles of the cell by little stringy things. I forget what they're called. I did not write them down. That probably would have been good to write it down. And then in telophase, new nuclei are forming, and in cytokinesis, the cell splits. So that's what mitosis is. Now, to make my brain happy, it's time for the history. Because everything has to be history with me. So cell theory began to form in the 19th century, which is actually the 1800s, because it's weird like that. There were most of it was German scientists. The main two beginners were Matthias Schleiden and Theodor Schwann, and they had the idea of free cell formation, which was like something crystallizes into cells. And this was the main theory for around 30 years, which really stunted cell research and, actually getting into things, because that's not how it works. But I understand where they were coming from, because cells are confusing. Then later there was 3 new scientists. I think one of them was French. I was really frantically writing down these notes because the video I was using, the guy was talking really fast, and I can't spell. So there's 3 new scientists. I think one is French and the other 2 are German. And they had the idea of binary fission, because they saw it happen with where is it? Eukaryotic cells. My handwriting is garbage. which is cells without nucleus. They saw them split, and they're like, huh, interesting. Let's write down that stuff. And then Walter Fleming, Antoine Schneider had some pretty good sketches, beginning ones. And then later, Walter Fleming he was like the one who really got the ball rolling. He used acids as a dye for better seeing abilities with stuff. I don't remember what the chemistry was, and I'm not good with chemistry, so I'm not gonna tell what was the specific things in it. But it's known as the Fleming solution. In 1879, he published a paper about indirect cell division in eukaryote cells. Chromatins mean the term chromatins, which are the half of the chromosome, means stainable, and it has ancient Greek origins, and it means stainable. He wrote a book on it. I didn't write down what the book was called, but you can probably look up Walter Fleming book on Google and find it. And I think another person was the person to do chromosomes, which means stainable bodies. So yeah, I'm tired. Goodbye. Look stuff up.